Hey guys, my name is Lucy Martell, and today I will be giving a presentation on CAR T Cell Immunotherapy. In this presentation, I will talk about what this immunotherapy is, how it works, and I'll include some data to demonstrate the success of the treatment. Specifically, I will give an introduction as to what CAR T cells are and how they relate to immunotherapy for cancers. Then, I will give a brief history of the research done thus far. Then I will discuss how these cells are made, how they work in the body, and how the treatment is administered along with some post-infusion effects. Finally, I will discuss the current and future research of this method of immunotherapy. There has been a variety of different areas of research regarding the targeting of cancer cells, but chimeric antigen receptor, or CAR, T cells have gotten most of the attention as of late. These cells are modified in a lab to target and destroy only cancer cells for a much more specific treatment as opposed to chemotherapy. Although there have been many published studies, the most promising results have been with blood-bound cancers such as lymphomas and leukemia. As of July 2017, the first CAR T cell immunotherapy was approved for commercial use. Novartis became the first company to get a gene-altering treatment approved by the FDA. Although this has been an incredible stride for cancer research, there are still so many things to optimize about CAR T cell therapy that it is still a very large and active area of research. Therapeutic antibodies have been researched since the 1950s. There has been a prominent amount of research with non-Hodgkin's lymphoma, in which clinical trials began in 1975 when there was technology available to create antibodies on a large scale. The early trials were done with rat and rabbit origins, which then moved to mouse and primate origins before finally using human antibodies to fight human cancers. In 1997, the first monoclonal antibody treatment was approved by the FDA for non-Hodgkin's lymphoma. Although the success started with B-cell malignancies, treatments have also been created for breast cancer, metastatic colorectal cancer, and melanoma. All of these treatments can work directly or indirectly to target and kill the tumor cells. Being as there are many options for cancer treatment, these methods can either be used as frontline treatments or as salvage treatments when the tumors are not responding to other methods. There have been many success stories with multiple types of cancer so far, but as every type of cancer differs, so must the treatment. There is still so much to discover in order to find the best treatment for every disease. The method for making CAR T cells is a long and difficult one. The T cells are taken from the patient's blood and allowed to proliferate for up to 2 weeks in order to create enough copies for treatment. The antigen receptor is induced via a viral vector that takes 2 weeks to create for a specific antigen receptor. This viral composition needs to be perfected before it's introduced to the T cells to ensure that the proper receptor will be coded for in the T cell. As you can see in the figure on the right, once the viral vector is perfected, it is put into solution with the T cells, and the two are mixed for 9 to 11 days. During this period, the RNA from the viral vector inserts itself into the DNA of the T cell and encodes for a change in the antigen receptor. After the 11-day mixing period, the solution is separated and the CAR T cells are put into vials and frozen before they're sent to the treatment facilities to be administered to the patient. There is limited research on the kinetics and destruction mechanism of CAR T cells on tumor cells. A study by Davenport et al. Investigates these issues using mice and a number of different antigen receptors to compare the kinetic data collected. In this study, they found that different antigen receptors have different kinetics. CAR T cells can destroy more than one tumor cell before they become inactive, and they also predicted that a combination of cancer therapies or a combination of 2 or more antigen receptors would enhance the killing ability of the CAR T cells. This is a figure from that study that compares 3 different antigen receptors, shown here as A, B, and C. This figure shows an increase of green, which is the calcium of the CAR T cell as it targets the tumor cell, and an increase of red, which is the propitium iodide in the tumor cell, which effectively kills the cell. Antigen A, in the top row, shows the levels of green and red staying the same since it was not a CAR T cell like B and C are, so the T cell was less effective at killing the tumor cell. Looking at B and C, you can see the tumor cell beginning to disintegrate and disappear as time goes on. This figure also helps to demonstrate the time difference in the reactions between the different antigen receptors. These findings allowed groups to continue research into optimizations of the antigen receptors to find the best type of treatment for different cancers. As I said before, the CAR T cells end up frozen in vials and sent to treatment facilities. In order to give the treatment, the CAR T cells need to be thawed out so they can be administered intravenously like most other cancer treatments. It has been shown that coupling CAR T cell immunotherapy with other cancer treatments, such as a lymphodepleting chemotherapy, increases the effectiveness of the T cells, just as the Davenport study suggested. A lymphodepleting treatment is a type of chemotherapy that begins to destroy the tumor cells and the environment so that they are easier to destroy for the CAR T cells. In a study by Dudley et al., it was shown that 51% of patients experienced positive results from both a lymphodepleting treatment and the CAR T cell treatment on melanomas. The pre-treatment included 2 days of cyclophosphamide and 5 days of fludarabine. Both of these medications are common chemotherapies that are known to lower the immune system and fight leukemia. As with other cancer treatments, there have been some side effects post-infusion that have been monitored and noted throughout studies. In a study by Ritchie et al., they aimed to test the safety of a CAR T cell treatment on acute myeloid leukemia. Herein, they show that there were a range of mild diseases both after fludarabine and CAR T cell treatments. These diseases were mostly containable and treatable, causing the group to determine the treatment a safe one. The type of disease and the overall outcome of treatment clearly differed from patient to patient, as it does with most other cancer treatments. Disease after CAR T cell treatment was present anywhere from 28 days post-infusion to almost 2 years. Remission is different with each patient, so this group was able to say that although there were relapses and stable diseases present, CAR T cell immunotherapy is a safe and viable treatment. Although this does not look promising in the table, this data is only from the individuals who suffered from illness, not from the entire trial. This study is evidence of another type of cancer that could benefit from this method of immunotherapy. Since blood-based tumors have been getting consistent positive results, the next goal for researchers is to get CAR T cell therapy to work on solid tumors. Solid tumors have a much different microenvironment than blood-bound tumors, so this has been proven to be a difficult task for scientists. There are teams working on mechanisms to control the CAR T cells after they have been infused in the body to lower toxicity effects. This is being done by teams who are making a type of switch to turn the cell on and off, and also by teams who are trying to provide the CAR T cells with nutrients for a longer lifetime in the body. The FDA-approved treatment is for people under 25 with recurring acute lymphoblastic leukemia, so the big picture is to get CAR T cell therapies approved for all blood-based cancers as well. The research done so far is beginning to revolutionize the way people think about and treat cancer, and we are lucky enough to be part of that generation.